Hi, everybody. It's your AP Biology teacher, Mr. Poser. Today, we're getting into topic 5.3, and that is on Mendelian genetics. So, yes, we are diving further into genetics, and what you can see on my screen here is a pea pod. Okay, so I thought this was on genetics. It's not about botany, pea plants, whatever. Yeah, peas are actually super important for the study of genetics. Why? Because they were the base organism here that one of what considered to be the father of genetics. He did some experiments with in order to determine how and why certain traits get passed down. So we're going to be applying a little bit of our knowledge from our last two topics, which were on meiosis, the production of sperm and egg gametes, and genetic variation. All right, the crossing over, the independent assortment, how everybody ends up different from one another, and how sexually reproducing organisms guarantee that their offspring are going to be different from each other and different from previous generations. Okay. Once we get a hang of like how and why certain genes get passed down from parent to offspring, we can start doing some science. We can start doing some hypotheses. And we can start making some predictions, and we can determine something called pattern of inheritance of a trait from one generation to the next. So let's get into it here. Here's a little bit more review from the last couple topics, a connection here. All right, you can see I have a set of chromosomes over here, and each of these chromosomes, I put a stripe on it and then a letter next to it. All right, these stripes are representing genes that are located on chromosomes. All right, and you can see that I've labeled each one with a either capital or lowercase letter. So let's look at these two at the top. They are both letter A, but this one's a capital A and this one's a lowercase a. All right, so these two chromosomes have the same gene, which is just A, right? But they have two slightly different variations of that gene. One's capital letters and one's lowercase letters. So these are two different variations or versions of that same gene. Same with B and C over here that are located on separate chromosomes. All right, so we're going to be applying some probability here, but that's a big if here, and we're going to talk about when this doesn't work later on. But as I put, if genes are independently assorted like chromosomes are, then simple laws of probability can be used to make predictions about their inheritance. Okay. Which means that we can use some simple addition and maybe some multiplication in order to make a prediction. Hey, if I take this organism with this traits and I take this organism with this traits and they have babies, what traits are their babies going to have, and how many of them will have this trait and how many of them will have this trait? We can do that kind of predictive power with some genetics and stuff that we're going to be learning about today. All right, a term that you might see a couple times here is what's called a locus. All right, a locus is basically the location of a gene on a chromosome. So if we're talking about A here, that might be referred to as a locus, right? Which contains a gene, even though it's got two different variations of that gene. All right, and as I underlined down here, patterns of inheritance can be determined with test crosses. All right, so the means by which a gene gets passed down from parent to offspring. All right, and how often that gets passed down from parent to offspring, that can be predicted using what are called test crosses. All right, so let's figure out what that is, a test cross. And we're going to start with probably the most famous test cross there is, but let me remind you of a few more things before we get into that. Okay, if we're sexually reproducing organisms, that means we have two copies of every gene. All right, that's what makes us diploid, right? We have two copies of every chromosome. We have two sets of chromosomes, right? Which means we have two copies of every gene. Okay. One inherited from mom, one inherited from dad. All right. So as a reminder, right, in human beings, the sperm cell has 23 chromosomes from dad, paternal, and then the egg cell has 23 maternal chromosomes. So when they fuse together, you end up with 46, two sets of 23 chromosomes in that zygote. And that zygote will eventually become a new, person if we're talking about human beings, right? And therefore, that new person has two copies of every gene. All right. So here's what's up. This point down here that says the different combinations of alleles, which are versions of the same genes, determine the offspring's traits. Okay, different combinations of alleles. All right, so which two genes that you inherit from mom and dad will actually determine your traits. All right, so check it out. What I put up here where I'm putting the same letter, just two different versions of that same letter. These are representing what are called alleles, all right? And you can get different combinations of these alleles. Maybe you got two capital A's and two lowercase A's, or two lowercase C's and two capital B's, whatever, okay? Those combinations of alleles are going to determine one's traits, all right? And this seems very abstract, so let's start from the basics. Let's go to the very, very beginning in the year 18 something. I don't remember what it is. But here's Gregor Mendel over here. He was a monk, right? And he was a gardener. He planted pea plants, and that was his hobby. You know, when you live in a church, I guess you plant peas. You know what I mean? One day, he's decided, all right, hey, I have these pea plants that have purple flowers on them, and then I have another set of these pea plants that have white flowers on them. All right, they're in two different parts of their garden. They don't interbreed with each other. So he's like, all right, well, that's interesting how this set of pea plants has purple flowers and this set of pea plants has white flowers. What would happen if, we crossed them? All right. And so what he did is something called cross pollination, right? You can, with plants, flowering plants like this, you can take the pollen from one plant and transfer it over to the other. And then you can take the pollen from that plant and transfer it back. Okay, and that's called cross-pollination. And basically, you can help these plants perform sexual reproduction just by, swapping their pollen pretty much. All right, and that's starting an exchange of genetic information that will produce unique offspring. All right, and then they're going to produce seeds that have a combination of the two parental plants. Right, it's pretty straightforward. Right, so he's like, all right, what if I take these purple plants that have only ever been in this purple part of the garden, and these white plants that have been only ever in this white part of the garden, and I crossed them? Well, what would happen? All right, we're going to call that the F1 generation. P stands for parent or parental. It doesn't stand for P this time. All right. This is a parental generation. All right. And what we're going to call the offspring of these two pea plants up here is what's called the F1. Now, make a prediction, just like Mendel did, just like a real scientist would. What colors do you think the petals of these offspring pea plants are going to have? All right. Are they going to be purple? Are they going to be white? Are they going to be like a nice lavender mix of purple and white, maybe a light purple? Are they going to have purple and white stripes, purple and white, I don't know, polka dots or something? Well, let's figure it out. Oh, well, they're all purple. Interesting. Okay. All of the pea plants from that generation have purple petals. And he's like, okay. Where did the white ones go? All right. Well, okay. What he did next is he took two pea plants from that F1 generation. Okay, and then he crossed those to see what would happen, right? He cross-pollinated those two plants. Don't think about it too much, okay? And when he cross-pollinated those, okay, we get what's called the F2 generation, or that's what geneticists referred to it as. What do you think happened in that F2 generation? Now do we get this mix of purple and white? Do we have purple and white polka dots? Are some of them purple and some of them white, etc.? Okay, well, he got a very distinct ratio here of the offspring. About 75%, or 3 out of 4 of the F2 generation plants had purple petals, and 25%, or 1/4 of them, had white petals. Okay, so if you've ever heard of a gene or a trait, say, skipping a generation before, maybe your grandma and grandpa had some kind of trait and you have it, okay, that trait skipped a generation. And he's like, I wonder why that is. So he did a ton more crosses like this and came up with some basic laws here. And they have to do with what's called dominant and recessive alleles. All right, what we spoke about before is that everybody has 2 copies of every gene, right? But sometimes some of those genes take priority over the other kind. Okay, they take they dominate the other one in others. In other terms. Okay, but as I put up here, using these 2 crosses, we can determine the pattern of inheritance for the purple flower gene and the white flower gene. All right, the purple flower gene, or what we call an allele, is dominant. Okay, only one allele is required from one parent to express the purple flower trait. All right, so all of these plants in the F1 generation are purple because this parental plant, okay, has 2 copies of that dominant allele. All right, so it gave 1 copy to each of these, and since it's dominant, that's all it needs to have purple flowers. It just needs 1 allele, and I'm going to show you what that looks like here in just a minute. Okay, and we can also determine that the white petal allele, the white flower gene, is recessive. Okay, and that's when it requires 2 copies of the white flower allele to express the white flower trait. Okay, so there are dominant alleles that only require 1, or excuse me, dominant traits that only require 1 allele, and then there's recessive traits that require 2. And white petals here is what's called recessive. It requires both a recessive allele from one parent and the other parent in order to get that recessive trait. And we're going to show you what that looks like here momentarily. Okay, so white is recessive and purple is dominant. Okay, and we're going to use letters here to represent the inheritance of those dominant and recessive alleles. All right, capital letters are going to represent the dominant alleles, and lowercase alleles are going to represent the recessive alleles. Okay, so if you have a pea plant here that has two dominant alleles, it inherited one purple allele from one parent and one purple allele from the other parent, guess what? That's what's called homozygous dominant, and it's gonna be purple. Okay, so I have a few terms that I've just thrown out here at you. The combination of alleles that an organism inherits is called its genotype. It's usually written as 2 letters or sometimes 4 letters. We're gonna look at that in a second, like it is over here. All right, so it inherited 2 dominant alleles. We use 2 capital letters, and that's gonna result in a purple flower. Now, what we call this genotype is homozygous dominant. It has 2 dominant alleles, all right, and these 2 alleles are the same. The prefix homo means same. Zygous refers to, well, zygote, right? This zygote has 2 of the same dominant allele, hence homozygous dominance, okay? But what about if you inherit 2 recessive alleles? All right, this plant over here got 1 recessive allele from one parent and 1 recessive allele from the other parent, so it's what we call homozygous recessive. Homo means same, zygous means zygote, recessive. All right, that zygote has 2 of the same recessive allele. What color is it? Well, it's going to be white. All right, it's going to have white petals. And then this last one, all right, what if you get 1 of each? Okay, you have 1 dominant allele and 1 recessive allele. Well, that's what's called heterozygous. Hetero means different. Zygous means zygote, right? So it has 2 different alleles. Now, here's what makes this dominant and recessive here. Ready? What makes purple dominant and what makes white recessive here is this, okay? When there's a dominant allele present, it's going to represent it's going to express what's called the dominant phenotype. All right, so these flowers are gonna be purple because they have 1 dominant allele. All right, the only way that they can have white flowers is if they have 2 recessive alleles, like over here. All right, the way we can illustrate this, and this is where it starts to visualize and starts to click for a lot of kids that I've taught before, is when we start using Punnett squares, okay? And Punnett squares, you probably did them in like 7th grade or something like that. And here they are coming back, and we're gonna go through the basics of them here today, in case we need it. All right, so let's do this P generation cross once again. All right, so this is the original purple flower and the original white flower. That Mendel cross is the P generation, all right? And since they only were separated in their, parts of the garden, we're going to assume that the purple flower is homozygous dominant, which means it has 2 dominant alleles, and this flower is homozygous recessive. All right, so how to use a Punnett square in case you need a refresher? All right, well, you bring each of these letters across, and these letters down, okay? And then you see what combinations they form inside the boxes, right? So here's the parents on the outside, okay? And then the offspring go in the middle. The offspring's genotypes, I should say, go in the middle, all right? So if you cross these 2 plants, all right, what we're going to look at here, we have 4 what are called heterozygous plants, and guess what? They're all purple, right? Because this dominant allele is present in each of these offspring, and that makes it purple, all right? And this explains why none of the plants from that F1 generation were white, because each of those was guaranteed to get 1 dominant allele, and therefore each of them is guaranteed to be purple, okay? And then if you recall, in that F2 generation, we had a different ratio here, all right? So here's 2 plants from the F1 generation, all right? So we basically took the kids from this generation, from inside this Punnett square, and we added them to the outside of this Punnett square. Okay, and let's cross them this time, all right? Each one of these are heterozygous, right, as we just displayed here. So I'm going to bring these 2 across. Once again, bop, 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 and bring these 2 down. And what are the combinations that we get? Well, check it out. We have this top corner plant is going to be homozygous dominant, therefore it's going to be purple. This plant is going to be heterozygous, once again, purple because of that dominant allele. This one's heterozygous purple. And then here's our 25%. Here's our 1 out of 4 that is recessive, that has a homozygous recessive genotype and therefore has white petals. Okay, check it out. There's a 1 in 4 chance of that happening if you cross 2 two hybrids or two heterozygotes over here. Hence, this is what's called a monohybrid cross. Each of the parents, all right, are a hybrid. They're kind of a blend of the dominant and recessive, and it always will result. A monohybrid cross will always result in a 3-to-1 or 75% to 25% dominant to recessive ratio. All right, so we're expressing only 25% of the white petals, which is our phenotypic ratio. All right, 3-to-1 would be. Okay. So yeah, there's the basics here. All right. Now something that you might be seeing here later on are pedigrees. Okay, and we can apply dominant and recessive. We can apply those laws to analyze a pedigree. All right, and we can make predictions. These are kind of fun, I think. You can watch the different kind of family trees and determine whether a trait in question is going to be dominant or recessive. Okay, so as I put here, patterns of inheritance, and then so far the only two patterns of inheritance that we talked about are dominant or recessive. There's going to be more that we're going to talk about later on, can be determined through pedigree analysis. All right, and when you're looking at a pedigree, if you see the square symbols on here, those are biological men, and the circles are biological women. Right, so here's mom and dad, and here's the kids. Right, they have 1 daughter and 3 sons over here. All right, so let's just say dad up here has some kind of genetic condition. Let's say he's got, I don't know, Tay-Sachs disease or something like that. Oh, that's kind of dark. All right, whatever. He has some kind of genetic condition. Okay, and then he has kids with mom over here, and then it looks like half of the offspring ended up with this genetic disease. Okay, so or whatever genetic trait we're talking about. All right, so what type of inheritance might that be? All right, well, let's look at this one before we can talk about that a little bit more. All right, so here now we have 3 generations. Here's grandma and grandpa, here's the parents, and here's the kids. All right, so if you trace this tree down here, check it out. Here's grandma and grandpa. The lines that go down from generation 1, all right, and attached to these squares and circles are, well, that's the direct line of that. Those are their kids, right? Hey, and then they're attached to, their partners over here, and then obviously the kids come from each of those branches. All right, so take a close look at this. All right, you got a key down here. All right, let's say some kind of gene or genetic disorder is labeled in red over here. So here's the affected male. Red circle is affected female. Blue square is unaffected male. Blue circle is an unaffected female. Okay, what do you think? Do you think whatever disease or genetic trait here is labeled in red, do you think it's dominant or do you think it's recessive? All right, well, let's take a look. Pause this if you want to try it for yourself. All right, I'm going to talk about it right now, though. Here we go. Here's grandpa. Grandpa has this trait, okay, grandma doesn't. And look, if you take a close look at this, all right, 1, 2, 3 out of 5 of those kids in generation 2 have that genetic trait as well. All right, and if you look at these 2 sets of offspring over here on the left and the right, the 2 sets of grandkids, check it out. This was passed down from dad to daughter over there, and it was passed down from mother to son and to daughter down here. Okay, so let's see. It was passed down, all right, and it doesn't look like it's skipping a generation or anything like that. So this is dominance, all right? And we can tell basically because if we did a Punnett square, all right? So think about this. Here's grandma and grandpa. If we did a Punnett square and we crossed big T with little t, or dominant T and recessive T, if we did that cross, what we'd end up with, if you try to work that out, is about 50% heterozygous, 50% homozygous recessive, all right? And 3-to-2 isn't exactly 50%, but it's pretty close, okay? So we can assume that this is going to be dominant over here, okay? And that furthers it here by showing us, hey, look at this was passed down from parent to offspring over here, and it's passed down from parent to offspring over here, all right? But check it out, this unaffected generation 2 daughter, okay, she didn't have any kids that were affected by it. It's only getting passed down from the ones that had that gene. All right, so therefore we can pretty much determine that this is going to be dominant. All right, pedigrees are going to get a little bit more complicated later on, but we're going to get there. All right, now we can apply this to 2 traits as well. Test crosses can involve 2 traits to determine their pattern inheritance. So we're getting back to pattern of inheritance. So we're going to get back to P's here for a second. All right, Mendel also, he didn't just do traits with like flower color and that kind of stuff, purple and white. He also, crossed plants with different that exhibited different traits, right? So there was some plants that had green peas and some plants that had yellow peas. There are some plants, that had wrinkled peas, and some of them had smooth peas. So here's the key that I'm going to show you right here. Peas can be green, which is dominant, or yellow, which is recessive. And they can be smooth, which is dominant, or wrinkled, which is recessive. Okay, so if we take a close look at this here, all right, to have green, smooth peas, okay, you need to be dominant for both traits. And we're going to use R and Y as the representative of the genotypes. R is going to be for the smoothness of the peas, and then Y is going to be for the color, right? So check it out. All of these genotypes are going to produce green, round peas, right? But check it out. The roundness is the wrinkliness, right, is going to be recessive, and the greenness is going to be dominant. So they have a recessive for one trait, but dominant for the other. And over here, the opposite is true. Okay, these are smooth peas or round peas, right, and they don't have wrinkles, so they're dominant for this R gene over here, right? But they are yellow, which means they're recessive. All right, and then this one down here is recessive for both. It is yellow and wrinkled. Okay, so what would happen if we crossed two heterozygous pea plants, right? Two plants that produce green round peas, but they are heterozygous. And here's their genotypes up here. We're going to do what's called a dihybrid cross over here, all right? A dihybrid cross, well, it's going to involve two different traits being passed down from parent to offspring. All right, and we need 4 times the squares for it too. So this is where it gets a little bit complicated, and you might be wondering where to begin. All right, but now we got to go back a little bit to our chromosomes, right, and our independent assortment. Okay, so if this is one of the parents over here, this is the genotype of one of the parents. There's 4 different combinations of alleles that they can pass down to their offspring that are going to be present in their gametes, in their sperm or egg cells. Okay, and how we can determine this is with the FOIL method. If you've taken, algebra 2 before, maybe you foiled different, factors and that kind of, factors, that's what they are. Whew, I haven't taken math in a while. All right, but anyway, you can use the FOIL method to determine possible combinations of alleles that can be passed down. All right, so there's 4 different ones from this parental genotype over here. All right, they can pass down, let's use F stands for first. They can pass down 2 dominant alleles, a dominant R, dominant Y. Outside, O, they can pass down a dominant R and a recessive Y. I stands for inside. They can pass down a recessive R and a dominant Y. And then they can also, L stands for last. They can pass down a recessive R and a recessive Y. All right, so there's 4 different combinations of R and Y that this genotype can pass down. All right, so what we're going to apply here are each of these genotypes, these combinations of these alleles. We're going to put those on the outside, and then we're going to cross them. All right, I'm going to invite you to try and cross these yourself right now and see what you get, because I guarantee you your AP biology teacher is going to make you do this at some point, so you might as well just get some practice in. But if not, I'm gonna press ahead here. Okay, but check it out. Each of these offspring are going to inherit not 1, but 2 alleles this time from their parents, okay? And there's a pattern here that's going to be pretty easy to recognize. Check it out. So if you put in the genotypes correctly, it should look something like this, okay? And you might notice something that's going on here. Now, which of these peas are smooth and green? Which ones are wrinkly and green? Which ones are smooth and yellow? And which ones are wrinkly and yellow? Let's figure it out. Ready? Okay, if you did this correctly, what you should end up with are 9 phenotypes with the green and smooth or round peas. You should end up with 3 yellow smooth peas, 3 wrinkled green peas, and only 1 wrinkled yellow pea over here. And this is what's known as a dihybrid cross. 2 heterozygotes for 2 traits produce a 9:3 to 3:1 phenotypic ratio. Okay, that's something that you can remember. If you see the dihybrid cross, the 2 heterozygotes being crossed like this, you can just remember, hey, 9:3 to 3:1, and you can skip this part next time. Maybe you work this out yourself, and if you did, I'm proud of you. But next time, now that you know it's 9:3 to 3:1 for 2 hybrids, you should be okay. All right. So yeah, dihybrid crosses can be used to make predictions as well. Now let's just recap here because this is going to be a long video. But if genes are inherited independently, then laws of probability can be used to determine alleles and make predictions about offspring. What we just demonstrated are called test crosses, and those can be used to determine what alleles or variations of genes parents and offspring have. The combination of alleles an organism has are what's called its genotype. An organism's genotype determines its phenotype, which are its physical traits. All right, so the combinations of A's or P's or T's or whatever that you get from located on the chromosomes determine your physical traits, which are called your phenotype. What combination do you get? Too dominant, too recessive, etc. Alleles can be dominant. The phenotype only requires one allele to be expressed, or recessive. Phenotype requires both alleles to be expressed. Genotypes are determined by what alleles offspring inherit. Okay, so just like we looked at the pea plants, the purple ones inherited two of the same alleles or one of each. The homozygous can be dominant or recessive depending on whether you inherit two dominant alleles or two recessive alleles, but heterozygous means you inherited two different alleles. And usually if you're heterozygous, you're going to express what's called the dominant phenotype, right? Because one dominant allele is present, and that's what makes it dominant. All right, and lastly, Punnett squares are used to represent test crosses and make predictions about the inheritance patterns of inheritance. All right, we also looked at pedigrees, and pedigrees can be, or Punnett squares and crosses can be applied to pedigrees to figure out the pattern of inheritance as well. All right, that was a long video. Hopefully you sat through it all. Let me know if you have any questions. We'll see you next time.